let me get started. Welcome back everyone for the second lecture of the day. After that brilliant lecture by Aditya, I don't know how I'll follow that, but let me give it a try. So I already gave my introduction in the morning, but just to reiterate, I am from Intel Labs. I'm a senior AI research scientist, and I also head the computational biology research at Intel Labs. And this lecture is going to be about, so the first lecture we went through the general basic concepts of LLMs. This is going to be more about how do we leverage LLMs in genomics and proteomics. So here's the agenda of the talk. We'll start with a brief background on LLMs. It's just going to be very short, just to reiterate some of the points. Then I'll follow that with why LLMs are appropriate for DNA, RNA, and proteins. Then I'll talk about LLMs used for DNA and RNA. What are the challenges and future directions in that? We can have a short break right after that, because that's a good break point. And then we'll talk about LLMs for proteins, followed by challenges and future directions for that. So we went through a lot about transformers and how they are used for language modeling. One thing which I think Aditya covered very briefly was the concept of pre-training and fine-tuning. So what basically happens is that for any task-specific thing, we may not have a lot of data. So what we first do is we learn a base model using pre-training on large amounts of task-independent data, and then we fine-tune that model on task-specific data. So even if the task-specific data is small, it still works out. So that's the general way of doing things in LLMs. And typically people use two kinds of pre-training techniques. One is a masked language model, and second is autoregressive language model. We saw quite a bit about autoregressive model in the previous lecture, where you have a bunch of tokens and the task is to predict the next token. But there could also be a masked language model where you have a bunch of tokens and you mask one of the tokens in the middle and then try to predict that. In case of masked language model, you need bidirectional attention as compared to autoregressive model where you need attention in only one direction. So some more background on the attention mechanism. So if you see this sentence, only one wizard could defeat Voldemort, Harry. We all know what Harry means here. Who is this Harry, right? But how do we know that? If the only Harry was mentioned, we would not know, right? It could be the Prince Harry also. But just because of the rest of the sentence, we know what it is. And that's possible because we have this attention mechanism which provides context to each and every token in the sentence. And of course, I'm taking some liberties here. The words are not exactly tokens, but I'm just taking the liberties so that it's easy to understand. So while attention is quite powerful and it allows us to provide context to each and every word in the sentence or each and every token in the sentence, it also has this drawback that the number of attention relationships is proportional to L square. If L is the number of tokens, or in the parlance of deep learning, it is the context length, the number of attention relationships is L square, as you can see here. Which means that beyond a certain L, it requires too much compute or too much memory. And the memory may not fit in your GPUs or even a large scale GPU cluster. And the compute may be it just may take too long for it to be feasible. So with that background, let's talk about why LLMs for DNA, RNA proteins. I think for this audience, I don't need to really do much of this, but just to set the context. So here's the central dogma of biology, right? We have genomics, we have DNA. That DNA is transcribed into an RNA. So that is the area of transcriptomics. Then it's translated to amino acid chain. And then that amino acid chain folds into proteins. And we can think of this as the DNA is like a cookbook. If I'm a chef, I have a cookbook. I can make any recipe today, but all the recipes are mentioned in the cookbook. Then I have picked a particular recipe to make today. That's like the process of translation or gene expression, where you get that RNA segment. And then once I have made that recipe, whatever is the final preparation, that's like the protein because that's the real action molecule. And interestingly, and I guess all of us know this, that all of these DNA, RNA, and protein, they can be represented over a language. So DNA, for example, is a language of 4 alphabets, which is what I'm showing here, ACGT. RNA language is just T replaced with U, so ACGU. And protein language has 20 letters. Because it's a language, you can think of a DNA segment as just very large text over this alphabet. And we are seeing a lot of LLMs are emerging that understand the meaning of these biological texts. So let's go through some of them. What kind of applications, let's just start from there. What kind of applications could this enable? So if suppose you have data sources, you have sequence data and you have some other non-sequence data also, you train an LLM. Suppose this is your LLM. You do the pre-training and you get this LLM. Then you do fine-tuning for various downstream tasks. And you can have functional region identification, so which are the functional regions in the DNA. You can predict which SNPs are responsible for which diseases. You can predict what genes would be expressed. You can even do mRNA sequence design. You can say that I want an mRNA which has these kind of properties, and you can do a design like that. You can predict the promoter or transcription factor binding sites for a gene. You can maybe predict what are the splicing or alternate splicing patterns. And if we are dreaming, we can even go to the point and say that what if my prompt was the whole genome, and I could just ask questions like a chat GPT. I can say, okay, which diseases are you susceptible to? Or what would be the drug reaction if I administer this drug to this individual? And things like that. So here is a sort of a tree of, or it's called evolutionary tree of various methods which are developed to build LLMs for DNA or RNA or proteins also. In this case, I have mentioned proteins also. And this is courtesy Jianma and Nicholas Ho from his group. They have built this whole thing. And this is a little dated. It's about a year old. So I will be talking about various models which are not mentioned here, which are more recent. But you can see that we have protein models and DNA models, and the DNA models can be of type encoder or decoder. Encoder would be that you pass a sequence and it will give you an embedding. And then you can use that embedding for various downstream tasks. Decoder would be it predicts the next token. So using that next token prediction, you can do a lot of things. So that's what this path is. And here we mentioned that some of these are using vanilla attention. But vanilla attention, as I mentioned earlier, has quadratic complexity, which means that depending on the length of the sequence, length of the context, it can take time which is square of that length, proportional to the square of that length, which may be a problem when we go to longer distances. And I mentioned that example in the previous slide where if you want to do the whole genome as a prompt, and whole human genome is 3 billion. There's no way a transformer can handle that. So then you need to figure out ways of doing subquadratic attention. And so there are newer methods which have subquadratic attention also, and I'll talk about all of those. So starting with this method called DNA BERT, which came out in 2021. So it basically pre-trains the BERT model. BERT is a regular large language model which was already available. They just took the same model but pre-trained it over DNA sequences based on human genome. They used for tokens, they used overlapped k-mers. So this is not some kind of learned tokens. It's just they just take the sequence and divide that into overlap k-mers. By that, what we mean is that first one will be AGC, and then you start with this G. So the next one will be GCA, and so on and so forth. So these are the tokens which are created. It allows for a context length of 512, and it is fine-tuned for various downstream tasks. And you can see here what we learned in the previous lecture. You have the original sequence, you create these tokens, and then you have token embedding and you have positional embedding. And you combine the two to create an input embedding. You pass it through a transformer. The transformer, so for example, on the right side, it says what is the classification. Is this clear, the figure? Yeah, okay. Because I'm seeing it from the side, it's not very clear. I'm not able to read those things. So this one is basically giving out probabilities for each token, each masked token. And then based on that probability, you can predict what the next token would be. So this is DNA BERT. Then came nucleotide transformer in 2023. It changed a few things. So firstly, it pre-trained on 3,000 human genomes. It took the 1,000 genome project sequences, pre-trained on them. It also pre-trained on several other genomes from 850 different species. It used non-overlapping k-mer tokenization, which means that for the same sequence as last time, where we got 4 different tokens, here you get only 2. So first would be AGC, and then you start from the A after that, ACT. And so they use this. This allows you to have a longer context, because as you can imagine in the previous one, each token only covers one additional letter, DNA letter. In this case, each token is covering 3 additional DNA letters. So it allows you to have a longer context window. And using that, they were able to increase the context length to 12 kilobases. And this has been fine-tuned for various prediction tasks. And I don't need to go into the details. It's basically the same process. Then came DNA BERT. And what they did was, so they realized that there are problems with the 2 tokenization schemes, overlapped or non-overlapped. If you use overlapped, the problem is that if I have these 3 tokens and I mask the middle one, I can actually recreate this middle one using these 2 tokens because they are overlapping with each other. So there is something called an information leakage there. Because it's so obvious, a large language model will easily learn that, and that would not be true learning, right? Then the problem with non-overlapped is that depending on where you start the k-mer, you can get a completely different set of proteins, different set of tokens. So if you start with this A and you create these tokens, then these are the token IDs. But if you start with the C after that, you get this set of token IDs. So it's just completely haphazard then. So instead of these 2, they use what is typically used in LLMs, which is byte pair encoding. And I won't go into the details of this, but basically it identifies patterns which appear more frequently. Like in the previous lecture, we were talking about the as an example, or many articles as examples which would appear more frequently, and it would create those as tokens. And they did a few more optimizations to improve the performance, and they were able to raise the context length to 36 kilobases. They also introduced, so until this point, the evaluation was, everybody was basically proposing their own way of evaluating their models, and they were showing good performance on their benchmarks. They introduced a benchmark which basically they made public, so people can compare on a standard benchmark.
So the challenge that you mentioned with the overlap k-mers, so that wouldn't matter if your goal is only to predict the next token, right? It only matters if you're masking the token.
Right, right. But both DNA BERT and nucleotide transformer and even DNA BERT 2, they are all masked language models. So for them, it made a difference. Okay, so far we have seen context lengths of a few tens of thousands, but we know that there are several applications which need very long context, and of the order of 1 million to even billions. So for example, the enhancer or silencer of a gene. So the way a gene expression happens is that there are promoters, enhancers, and silencers, and they act in different ways on the gene to sort of regulate its expression. And they can be quite far also. They can sometimes be a million letters away, million bases away. So in that case, a context length of tens of thousands will not really help you. So you need much larger context window. Also, there are many of these other applications which are sort of similar. So if you want to predict variant effect, which means that if I change a letter in the promoter, how will that change the gene expression? It's the same problem. The promoter is too far, right? And similarly, many others here. Also, another problem you can think of is that 3D chromatin organization prediction, where, as you guys know, the DNA is actually coiled into chromatins, and those chromatins are actually pretty long sequences. So if you want to predict how they will be organized, it will not be possible with a small context window. And suppose you want to now design a full chromosome from scratch. A chromosome is hundreds of millions of bases. There's no way small context window will be enough. And my favorite example from earlier, if you want to do the whole genome, again, same problem. So how do you do that? And the other problem we are facing is that each nucleotide matters. So for example, look at this. This is about sickle cell anemia. If you see normal DNA, this is how it looks, and this is what the cell looks like. The only thing that has changed is one letter here, and that has led to sickle cell anemia. So every single letter matters. So whether you do bite pair, whether you do k-mer, overlap, non-overlap, you may lose that information of a single letter. So you would want to have tokens which are individual nucleotides or individual bases. At the same time, you want to support a very long context window. So you need newer methods which can basically increase the context window quite a bit. So the first example of that effort is something called hyena DNA. So if you see here, this is our attention mechanism where we have an all-to-all. So for each letter, it depends on all of these, and hence the computation required is order n square. What they did was they replaced the attention with something called a hyena operator, and where what they did was, in addition to a bunch of small convolutions, and I won't go into details of that, they also added something called a long convolution. So long convolution takes care of longer context window, but again the problem is that a convolution operator which has if the size of the convolution operator is same as the context length, which is what we want, then it can still be order L square, because again for each letter here it depends on L letters at the top, so it can still be order L square. But what they did was they used a mathematical formulation which says that you can do the same thing in FFT domain, and in FFT domain that n square becomes n log n. So that's how they've improved the computational complexity. Also they use auto-regressive model. So earlier models were more mass language models, the later models are auto-regressive. They tokenize at the nucleotide level, and the advantage is long context length, so their context length they can support is about 1 million, and they are significantly faster. So at the 1 million level, they are 160 times faster than the fastest implementation of transformers. Disadvantage could be that it's not clear if convolution can match the power of a transformer, right? The reason we moved to transformers from convolution was to gain more power, but yeah, so that is one potential pitfall. Then to take care of the problem that convolution may not capture everything, a new model was proposed, which is, so previous one was hyena, this is called striped hyena architecture. And the idea of striped hyena is that you have these hyena operators which I described in the previous slide, but you also have a few attention layers. And with those few attention layers, you hope that you can capture the longer context window at good accuracy, and with hyena you do the rest. So for example, this language model called EVO, it has 29 hyena layers and 3 attention layers. This also tokenizes at nucleotide level. It can go to a context length of 132, and this seems regressive, right? Previous one was 1 million, now we have 132. The reason for that is that because we also have a transformer architecture there, we cannot have much higher context length. This was trained on prokaryotic and phage genomes, and it also demonstrated, this was the first model to demonstrate that a foundation model or a model LLM learned on DNA is able to learn aspects of protein and RNA sequences. Because protein and RNA are derived from DNA, one would think that that is possible, but this is the first model to actually show that. After that came EVO 2, next version of EVO. And here they made a few changes. So instead of striped hyena, they now use striped hyena 2 architecture. And it has slight differences from the striped hyena. So striped hyena is the one at the bottom, striped hyena 2 is the one at the top. And you can see that here we have similar hyena operator blocks, but here we have 3 different types of hyena operator blocks. So the 3 different types are basically short convolution, medium convolution, and long convolution. So they have divided that into these 3, and they also still have self-attention layer, and they still do nucleotide level resolution. And now with this, and I guess because of improvement in GPU architecture, they were able to get to the context length of 1 million. Striped hyena is also trained on very large, sorry, not striped hyena. EVO2 is also trained on a very large amount of data. In the previous lecture, we heard anything which is 10 trillion or up thereabouts is what is considered modern. So it is trained on 8.8 trillion tokens or 8.8 trillion nucleotides. And these are coming from bacteria and various different kinds of organisms. The whole thing is listed here. Each point here is actually a genome. So there are very large number of genomes here and very large number of tokens extracted from them. And there somewhere it's mentioned, human is also mentioned here. So there is human, they even have gorilla and several other kinds of organisms. They used a two-phase training strategy to sort of make the training more feasible. If you were to go for 1 million context length throughout the training, then you have to do a lot more compute. So to avoid that, what they did was they did first pre-training with only 8K context window, and then they further pre-trained, so they are calling it mid-training, but it's basically another kind of pre-training. So they further pre-trained with 1 million context length sequences to get to that 1 million thing. So EVO actually is a fairly impressive model. It learns from DNA sequence alone to accurately predict the functional impacts of genetic variation without task-specific fine-tuning. So in the previous ones we saw that we have pre-training and then task-specific fine-tuning. The better your pre-training model is, the less fine-tuning you would require, right? So this one shows that just by pre-training it was good enough for many task-specific, many different kinds of downstream tasks. Also, it autonomously learns the breadth of biological features, including exon-intron boundaries, transcription factor binding sites, etc., etc. It is able to generate various different kinds of sequences, so mitochondrial, prokaryotic, eukaryotic at genome scale. So hundreds of millions of bases with greater naturalness. So they are very natural and coherence than previous methods. So I just have a table which summarizes all those which I have detailed in the previous slides. We don't need to go into details of this. Only thing we need to see is that it seems that EVO2 is the most impressive model of all in this list. It is trained with the largest number of parameters, so 40 billion parameters here. It has a pretty high context window. No other model has a higher context length than this one. And it shows good performance on several different kinds of tasks. But this table is very interesting in my mind. So we went through various categories, right? We did transformer first, and then there was hyena. There is something called mamba also, which I don't have the time to get into details of, but it's another kind of sub-quadratic method of training, which has shown to be highly promising. And it is being used in a couple of DNA language models, and it performs fairly well. And then we have these hybrid models, which are basically either combining transformer with hyena or transformer with mamba. So we saw examples like EVO and EVO2 of those. There is hybrid DNA also, which is mamba plus transformers, etc. And they sort of combine best of both worlds, so transformer or the subquadratic ones. And they have efficiency of long context length with fine-grained details, and they generally achieve SOTA performance or state-of-the-art performance on very diverse range of tasks. But SOTA doesn't mean problem is solved, right? So now let's talk about promises, challenges, and future directions of LLMs for DNA. So while foundation models for genomes have shown to have potential, they have shown promising results, pre-trained models are easily adaptable to multiple tasks. We have also realized that biology is fairly complicated. Its language is likely harder to model than human or natural languages. Biological data involves many confounding factors. I don't know if we are covering all kinds of confounding factors if you're just focusing on the language or the language modeling. It is extremely high dimensional. Many times the questions are also not mathematically well defined. So do we even know what are the best questions to ask? And I'll elaborate on these points in the later slides. So let's start with the first challenge, how to be more foundational. So I mentioned earlier that if your pre-trained model, which is also called foundational model, if that is trained well, or if that is powerful enough, then you need less of fine tuning and less of task specific training, right? So while we have, so the foundational model has to be good, right? But what we have seen is that although trained on trillions of tokens, current foundational methods, they struggle with zero-shot settings, which means that you cannot just apply them to different settings without going into details of zero-shot. We also see that it fails to separate biology from noise and sometimes has no effect. So for example, there was this study which said that we found that genomic language models representations provide little or no advantage compared to standard one-hot sequences. And now we all know what one-hot sequences are. So what they did was they took the foundation models which were available. So you can see this is tokenization, then you have a BERT-like model, mass language model. So this is the foundation model. And that will give you some embeddings, right? Some representation of the data. Instead of that, if you use just one hot representation of the same data. So AC, GT are 4 things. So you can do one hot representation where you have 1 and 3 zeros for each of them. And now if you compare the two, so what they did was they trained a very shallow convolutional network based on these two. It was deliberately kept shallow because they wanted to make sure that what we are learning is from the foundational and not from that network. And they tested it on several downstream tasks. And I'm just showing example for one of them. So this is alternative splicing or RNA elongation. And here what they see was this one hot one is actually performing the best out of all. And even here, one hot is not the best, but it's not too far behind the foundation models. So are we really learning anything here? So, but does that mean that we need more training data? Because as I'll show in the later slide and what Aditya also mentioned, there are scaling laws, right? More data, more powerful, bigger model, it generally does better. So does that mean we just need more training data, or we need different modeling approaches to make the models truly foundational? And the other thing is, do we have enough training data? Because human genome is highly diverse. Do we have enough data to cover all kinds of structural variations, all kinds of SNPs and all that? Second challenge is that not just the foundational, when you do task-specific fine-tuning, it does not always outperform simple specialized methods for those tasks. So it's like jack of all trades, but master of none of those tasks, right? So questions are, how do we assess the effectiveness of a strategy? Like if we are designing a model, how do we assess the effectiveness? Because we don't want to be down millions of dollars of training cost and then not really get anything out of it, right? And how can we better harness the power of these models? Third challenge is, where are the nails? So you guys may have heard of hammer and nail analogy, right? So foundation models are powerful hammers. There's no doubt in people's mind that they are powerful, but they are not being able to show the effectiveness. So surprisingly, it seems many biological questions do not even look like nails. They don't even need that kind of complexity. But there may be others. So many methods, they are excelling in supervised tasks, but genomics is also mainly exploratory, right? We don't even know what questions we want to ask. So what exciting biological discoveries can foundation models enable? Do we know what questions to ask? Can a model answer a question that it was not trained for? Can a model uncover unique characteristics without even predefined questions? So I would like to show like a couple of things here to properly make that point. So suppose we have, this is a sea of all the questions to be asked. There is a pool of questions which an LLM can answer. There's a pond of questions which biologists have. We want to find out what is that intersection. And I don't know if we know that. And another way to think about that is in terms of knowledge quadrants. So there's this concept where we divide the knowledge into 4 quadrants. I know what I know what I don't know. But it's possible that I don't know what I know. Sometimes I may surprise myself that I know some things, but I wasn't aware. And the last one is the trickiest of all, which says, I don't know what I don't know. So you may think you know a lot of things, but it turns out that we don't. So we don't even know what questions to ask. So we don't know what we don't know, and hence what questions we have to ask. Next challenge is, do we need bigger context lengths? So why? Why would we need that? What are the applications? So I've already covered this, so let me skip this part. and how do we achieve longer context length given the compute available and without compromising any accuracy. We already saw multiple approaches, but maybe something that is missing is, can we use additional information we already know about interactions in the biology domain? Maybe they are not there in the language domain, a natural language domain, but in the biology domain, maybe we know more about how the DNA elements interact with each other, and maybe we can use those to figure out what portions of the DNA we need to attend to, or we need to have attention to. Next challenge is interpretability, and this is very important in biology. How does an LLM work? Frankly, nobody knows exactly. We know how it works. We went through an entire lecture, but the point is that we don't know what is in those giant weight matrices, what is there, and if we say that, so in the biology context, if we say that this particular gene is responsible for this particular disease for an individual, and if you take this drug, then I know that this problem will be solved. Can we actually see that in the in the workings of the LLM? Can we see that in the weight matrices, that this is how it is reasoning to come to this conclusion? If we don't see that, we already know that there is data is biased, data may have noise, so the data may be corrupted. We also know, we don't know how the LLM is working. It's just coming up with one final thing that if you take this drug, you'll be fine. We don't know how many takers will be there, right? So adoption may become very, very tricky if we are not able to interpret the language models. And this is an active research area. In fact, people are developing some visualization tools which can aid interpretability, but it's not a solved problem yet. And I have a bunch of more challenges. So how do we handle unique noise in biological data? How to integrate multimodal data, DNA sequences, RNA counts, proteins, spatial information, prior knowledge in terms of what we know about humans or other organisms. Also, LLMs have a huge number of parameters, sometimes requiring billions of data points to train. Often there is insufficient task-specific labeled data. So what do we do about that? It requires significant compute, which is not approachable for many academic labs. So how do we get around that? There are data privacy and security issues, and there are biases in data and models. So with that huge list of challenges, let's see what the future can be like. So the first question, natural question would be, do these models truly have a future if they're struggling to find biologically meaningful representations during pre-training? Why should we interpret, and why should we try to improve these models? So first thing is that they have significantly higher ability to model the underlying patterns versus task-specific or other traditional non-LLM based methods, right? They have significantly more power, and the bigger the model, the more power it has, right? So we have seen in scaling laws, and Aditya was referring to that, what we see is that if you increase so the y-axis is the test loss, and lower the better. The x-axis is various things here. So if the x-axis is compute, as you increase compute, as you provide more compute to the models, you actually reduce the loss. You provide more data size, you reduce loss. You have a model with a larger number of parameters, again you reduce loss. So far this has been happening, and we still haven't seen the tapering yet. So who knows, maybe if we just keep having bigger and bigger models, we are able to solve this problem. Another thing is that they are generative. What does that mean? That you can generate sequences once you have learned these models. This is not possible for other previous traditional methods. So another reason to invest in these. And also, just because the models currently aren't leveraging the full benefits of pre-training doesn't mean that they can't. That just means that maybe there's more work to do. In fact, there was this paper, it's a little old, it was published in 2016, but it talks about future is unsupervised. Right, so there's no way we can have labeled data, and all of those things may not be possible. We have to figure out how to do this in an unsupervised manner. And LLMs are prime examples of how that can be done. And so the other question to ask is how to build general purpose model to answer unseen questions about a data set and to generate hypotheses and ask new questions. And again, we don't have answers, but general consensus is that the next frontier of large language models is biology, and we have the 3 Nobel laureates here as a, as a proof of that. So today, while today we are at this point where we say to a chat GPT or whatever, to an AI chatbot, write a piece of code in Python to do so and so, or we may ask questions about a certain disease, or we may ask it to summarize a paper. We are working towards getting to a future where we can say generate DNA sequences with these desired functional properties. And once we are able to do that, we can engineer biological systems. So the possibilities are endless, right? Or we can say that here is a data set, find new knowledge for me. I don't know the questions. You just figure things out and do it for me. So all of these things may be possible in the future if we invest in this. So that was LLMs for DNA. We can do a short break here if you want, or if you want, I can continue also. What do you think? I think we can continue, right? Okay, so no break then. Okay, so LLMs for protein analysis. So as we all know, proteins are action molecules.
Maybe we could pause for questions.
Yes, yeah, oh yeah, yeah. That's a good idea. Any questions on LLMs for DNA before we move to a different topic? I think it's a combination of both. So I think Mamba-based architectures may also do very well, hybrid Mamba plus transformer-based architectures, and Evo 2 is not that. Evo 2 is Hyena plus transformer, but they haven't been trained on such large scale yet. So if that happens, we have to, like, it's there to be seen whether they will be able to match the performance of Evo 2, but that's currently the best model out there. Mm-hmm.
That's sort of the next one.
Mm-hmm.
In this setting, how would you compare these tasks to what a trained human could do?
Trained humans cannot do if you can't read hundreds of millions of ACGT and figure things out, right?
Given enough tools and enough time, it's not possible.
With tools, maybe it is possible. Like if you develop, I think you'll need maybe several years, but if you develop that domain knowledge of that specific task, and then you have tools to do this, maybe you'll be able to do it.
It seems like the bar is already
Yes.
for everything.
Yes. Yeah. Can't hear you very well.
Okay, so basically there would be overlapping diseases, like a similar disease set of genes and all would be at play. But it is very difficult to put a, like demarcation, like, okay. Because usually when we talk about biomarker discovery also, we say so-and-so biomarkers are for this disease condition A, so-and-so biomarkers are for disease condition B. But
and
when
they will be common between the two.
Yes, they will be common. So how do we, because, and sometimes what happens is in people, there would be more than 2 diseases also. So in that context, how can we properly demarcate this?
I'm not sure I'm getting the question, because what do you want to demarcate? Ultimately, for an individual, we'll be predicting whether this disease is, whether this guy is susceptible for this disease or not. And the same question we'll ask for the other disease.
Yes.
Both of those questions can be answered through, if LLMs are able to train on the entire genome, they can be answered.
Yes.
Right. So I'm not sure if I'm getting the question.
No, like I mean, it shouldn't be like, say suppose I'm having both metabolic and this, say suppose another disease is there, then the LLM model should say you have both this and this, not like 50% this and 50% that.
Right.
So, but it could also happen because of there are overlapping genes, like because ultimately there are genetic path, like there are pathways, and there are some common factors which come there, right? So, do you think LLM would be able to distinguish the differences between these overlapping diseases? Because that is one of the problems which we face with metabolic disorders. I mean, some sort of I'm also working on sort of thing, and I find a lot of overlaps, which is making it very difficult to find a demarcation. So do you think LLMs would be able to solve that?
Actually, I'm still not sure if I've got your question, because if I'm thinking in your domain, if you are not using an LLM, suppose you have these biomarkers and then you have some other machine learning model which is predicting individual diseases. So that machine learning model, either you are training it for like you are asking the question, is this disease there or not? Then it should be straightforward. Or are you asking the question, what all possible diseases can be there?
What all possible diseases?
So in that case, it's possible that you need a multi-class prediction algorithm that may be able to handle that, right?
So what about like LLMs, like specifically for because
I think LLMs would be more powerful to be able to do that job. Whether they can completely do that job or is there to be seen.
Okay.
I don't know if that answers your question.
Okay, sir.
So if we are using existing pre-trained models and we are getting normal accuracy like 80, 85, something kind of, I need to increase the accuracy. So what kind of changes that we have to do, sir? Like either we have to increase the training data sets or completely change the pre-trained model architecture, something kind of, like what are the aspects we have to search or we have to think?
Right. So there are two ways to do this, right? On any task specific, like if you're looking for a specific task, either you keep the model the same, pre-trained model the same, and you train it on more of task specific data, right? Which is, I'm assuming that it will be labeled in some way. So you can use that. Or you have a bigger foundation model, so you train the pre-trained model on a much larger amount of data, and hopefully that will cover the relevant aspects to be able to do this better in this way.
Only these two ways, sir? Because while we are going for the task-specific classification or prediction, the amount of data that we get is very minimal. So for doing the fine-tuning or pre-train, fine-tuning the model basically.
Right.
So in that case, when we need to change the hyperparameters in order to tune the data that whatever we have, what are all the other things that we can do, sir? Other than this.
So there is also an active research in developing synthetic data or even generating new data, right, which can sort of augment the lack of or elevate the problem of lack of data for task specific purposes. So you can do that, like you can basically think of various scenarios and you can generate data for those various scenarios and then train an LLM to do that.
So one more thing is, like you have shown in one of your slides, like the promoter, enhancer and silencer.
Yeah.
Say suppose one of the objective that I'm focusing is to predict the silencer or promoter regions. So even if we trained the task specific sequences for the training and testing, the one thing when we are validating the sequences, like whatever sequences that we give, whether the model says that is a promoter or a non-promoter, the tasks after prediction, like we have to validate, somehow we have to state that this is the reason the model says that it is a promoter or non-promoter, silencer, non-silencer. So for that aspect, how seriously we can't go for the experimental, so since that is costly and most of things are not possibly there in labs. So how to handle that scenario, sir? Like how to justify the predictions given by the model, LLM?
That's the interpretability problem, and there's no solution to that yet.
Okay, sir.
Yeah, I wanted to ask about the variant calling and variant interpretation part that you talked about. So currently how we work is that when we use a variant caller and we say that this is an SNP or a SV, we have certain evidences associated with it. So for SNP, this is the leak depth, for SV, the coverage is low or whatever like that. So what I wanted to ask is, can LLMs provide evidences along with claims that this is a variant or this is a structural variant, and to make it more kind of believable to us? Because like even in current scenario, if we find certain variants or structural variants, if we do not have that level of evidence, we discard them as false positives. Can that be done using LLMs?
So let me ask you, what kind of evidence would convince?
So as I said, say for example, in structural variations, we look for split reads, what is the distance between them, coverage, how it is decreased or increased, things like that. Can LLMs come up with these evidences?
I think it's possible. So if I understood your question right, if suppose LLM says that this is the variant and at this particular position,
Yeah.
Can you go and see that?
Yeah.
Right? So if it's giving you the position, and if it's and I'm sure an LLM can even print the part, that particular portion of the DNA. It can print that one and the reference sequence. You can see for yourself whether the variant is there or not.
I want to know what evidences it has considered to call it a variant.
That is again the interpretability problem, right? So, by the way, for interpretability, there is at least I'm aware of one professor who's working on this, and his work I respect in this field, Anshul Kundaje. So if you're interested, just check out his work. He's done a bunch of things in the interpretability of deep learning model architectures. Maybe there's something interesting there.
Okay. Thank you.
So there would be, in the context of information we have, there could be some arenas we might be lacking information itself, like some rare diseases or like which happens to some people only, and we have less data also. So in that case, we are competing like that is a small sample size, and the other, say for example, cancer, which is very fairly common, and all, we have larger sample size. And in context of constructing such a disease specific model, like how do we combat the smaller sample sizes data with smaller sample sizes, because that is all we have, right? And again, we can say like, okay, interpretively it can be biased, but like, I mean, it could be overlooked, but how can we solve, like we should have some sort of mechanism for these small rare diseases also. So in that context, what how can LLM
So in my mind, yeah.
Perhaps it would be helpful also when we talk about these problems to talk about what the task is or what the input data to the LLM might be. That might help get everyone on the same page.
Right.
So in this case, what your task is to predict if a variant causes is responsible for a rare disease, right? What would your input data be? Like, what are you training the model on? Oh, sorry. Just pass it.
We already have genomics and like suppose we have only multiomics data. So we consider like we have multiomics data, but what happens is like the ideal situation is that say suppose there are 40 people in this whole world which is having a rare disease. The ideal situation is we have the genomics, proteomics, metabolomics, all the sort of multiomics data, and then we build the LLM.
For all 40 of them?
Yes, all 40 of them. That is the ideal situation. But more or less it doesn't happen. And things change because transcriptomics is dependent on the activity, like it changes time to time, and that's like a snapshot, like you said. So so in that case, we have a lot of missing data in that scenario. So say suppose we consider multiomics data here, like NGS data. So it's how can LLM leverage those? I mean, leverage its architecture and resources to make better interpretable models in this scenario?
That's a very difficult question, I guess. So I'm aware of, so there used to be one project at IGIB where what they would do is for rare diseases, they would go and do sequencing themselves to increase the amount of data available for that particular task. That is one approach, but you're saying that if that is not possible, then what do you do? Another possibility is, like I was saying earlier, that if you know what kind of data you generally get, you can probably increase that data by synthetic ways. But if you are not aware, or if your synthetic data is not representative of the real data, then it doesn't really help there, right?
Usually synthetic data will take the lowest and the highest. Sometimes it happens like it will take a range of the data and then give us the synthetic. Like sometimes it happens with smooth also.
No, but that is if you're not using an LLM. If you're using an LLM, maybe it can generate that synthetic data for you.
Yes, but how much can we rely on the synthetic data that will suit my
Right. So I'm not sure if you want to take that question.
I don't know the answer for specific for rare disease. I know there are, as Sanchit said, there are now companies that are trying to, you specify a issue of interest, specify a condition, specify a population, it will generate your RNA sequencing data set for you. I have not tested these myself to know how accurate they are. I suspect it will depend on the severity of what you're looking for, right? If you're looking for something that's not changing a lot of biology, perhaps your synthesized data set will be fine. It's actually an interpolation problem, right? That will be okay, but if you're testing a major change, I don't know how.
So it will be safe to say that we have the half of the data which we need actually. Like I mean, we need more data so that in the coming years, if say suppose people are going towards this personalized medicine and they get sequencing, then we would get like big projects. From the big projects only we would get more data and thus the input for the LLM.
Right. I think there are only three ways of thinking about this, right? LLMs are going to be are models which are trained on data. If data is not there, they really cannot do much, right? The only thing that is possible then is that if LLMs have just learned the overall language of DNA, suppose it has seen enough other data that it has learned the overall language of DNA, maybe it is able to produce synthetic data, and then that you can use that for your purpose. But otherwise, if certain kind of structures or certain kind of features are not present in other data and only present in 40 individuals in the world, then LLMs would not have seen that kind of data, so it will not be able to produce synthetic data for that, or even reason about those things. So I don't know if there's any way around that.
Sir, actually there are encoding methods for particular composition of or given sequence of subset of DNA. It has a particular physiochemical property of the DNA, like tilt, twist and all. So like, is it possible, I'm not aware of much of LLM architecture, but is it possible to artificially give tokens, like a particular subset of sequence is responsible for this physiochemical property of DNA, or is responsible for promoter or binding site? Like there are descriptors that take in a DNA sequence, and then they give the kind of, it's kind of a calculation based thing, it gives the physiochemical property of the DNA. So is it possible to tokenize this information for large language models?
So let me see if I've understood your question.
Okay.
Are you asking that given a sequence,
Yes.
is LLM able to predict whether it's going to be a promoter or whatever? Is
No,
that
sir.
the question?
Actually, there are particular descriptors that take in a sequence and give physiochemical, based on physiochemical properties. Like
Descriptor meaning, is that a tool?
No, sir. Like particular, for example, in protein, particular amino acid is responsible for hydrophobic. If it is spaced in a particular way, it is responsible for particular helix or like loop and all. So that information, can it be tokenized so that LLM
Yes.
can?
Yes.
Okay. So what kind of models currently are implementing these techniques?
See,
LLMs?
the model does not really. Okay, so if you're talking about alpha helix and beta sheets and all of that in proteins,
Yes.
then AlphaFold does all of that.
AlphaFold.
And I'll talk about that in the later slides.
Because as I understood, only based on raw patterns, it's trying to understand whether it will become a helix and all. So
Yes.
if possibly we can introduce some information from our learnings.
Yes,
Okay.
absolutely. In fact, you can design your own sequence
Yes.
and ask AlphaFold whether what kind of structure it will produce,
Okay.
and then you can see whether what you wanted has happened or not. Any other questions? Okay, let's get to the next part. Okay, so proteins are action molecules, and I really like this quote by Arthur Lesk. in the drama of life at molecular scale, proteins are where the action is. So proteins are involved in virtually every important activity that happens inside our living thing, digesting food, etc. I don't want to go into details of this because everybody knows here. Proteins are so important because they are so versatile. They can undertake a vast array of different structures and functions, far more than any other type of biomolecules. That's why proteins have been so successful and have been so important. So what are proteins? They are linear sequences of amino acids that can fold into a complex 3D structure. A protein 3D structure, protein 3D structures define their functions. For example, hemoglobin is the oxygen carrier, right? And it's because it has a structure like that which can bind to an oxygen molecule. And there are 20 standard amino acids. So since the protein function depends on its structure, we would like to know what that structure is, right? So that we can do more with the proteins. The situation is that getting a protein sequence is fairly cheap. You can, for example, sequence the DNA, get the corresponding RNA, it's a one-to-one correspondence, and get the corresponding amino acid sequence, again one-to-one correspondence, fairly cheap. But experimental discovery of the 3D structure of a protein is very, very expensive, and you can see this is a whole process here, which is X-ray crystallography based. You have to express the protein in a cell, then break open the cell, purify the protein, etc., etc. A long process, it may take years and millions of dollars per protein. So there is a problem to be solved here, and hence AI comes to rescue. So we can use AI to predict the 3D structure of protein directly from the amino acid sequence and bypass all of these steps. And of course, we should know that experimental methods are going to be more accurate, but for many scenarios, we don't need that level of accuracy. So for example, if you're a protein design expert, you're designing various different kinds of proteins, you just want to design something and see whether it is going to be similar to what I'm expecting or not. You may not want to do this cryo-EM thing for every single design, because if after every design you have to wait for a year, it doesn't make sense, right? So there are many scenarios where just doing a simple structure prediction using AI would be more beneficial. So what are the challenges? The 3D structures are complex. So for example, here if you see, this is a primary structure, which is a sequence. Then we have secondary structure where we have alpha helix and beta sheets. Basically, these sequences have folded into these shapes. And then we have tertiary structures which has multiple alpha helixes or multiple beta sheets, and they are connected in various ways. And we even have quaternary structures where multiple proteins are combined together in a giant structure which is called a protein complex. The bigger the protein, the more difficult it is to model, and the space is astronomical for the 3D structure. So AlphaFold has been fairly successful, as we all know. So there is this competition called CASP, which is a competition for protein folding methods. It is organized every 2 years. Teams predict the structure of newly experimentally discovered proteins. So the proteins which have been discovered over the previous 2 years, those structures are not made public, and teams predict those structures, and then they are verified against the experimentally discovered ones. This is the measure of metric of success. It's called global distance test. It ranges between 0 and 100, and if you get a score of more than 90, it is considered a very competitive, and it's considered that the problem is solved. So you can see here, AlphaFold 1 came in 2018, and it got 60%. But then AlphaFold 2 came and it got more than 90%. It got a 90, not percent, but a score of more than 90. It got a score of 92.4, which is when it was considered that the problem was solved. For many cases, it's not solved for everything, but for many cases. Impact of AlphaFold, it's considered the most important achievement in AI ever. Of course, these hyperbolic statements can be disputed by various people, but it is definitely one of the most important achievements of AI. It is a giant leap for biology. It solved a 50-year-old problem. Nature called it the method of the year when it came out. And it also won the Nobel Prize in Chemistry in 2024, which is testament for its impact. And you can see that number of papers which are citing or using AlphaFold are increasing at a rapid pace. And this is dated. This is 2022. Now this is going to look much, much bigger. So what is the AlphaFold architecture? And this is sort of different from the LLM kind of architectures, which LLM for DNA architectures, which we saw earlier, because it has non-deep learning stages as well. So AlphaFold, what is the problem it's trying to solve? It takes a protein sequence as input and it predicts the 3D structure. There are three major phases: pre-processing, which is non-deep learning, and then we have evoformer, which is a transformer-based, so it stands for evolutionary transformer, and there's a structure module. So let me go to the next slide. Yeah. So what does pre-processing do? So what we do is, if this is our input sequence, we do a genetic database search. So this is a database of all proteins known to man, and it looks for proteins which are very similar to this input sequence, and it stacks them together here. The idea is that proteins which are very similar to in sequence will perform a similar structure, which means that they're evolutionarily conserved, which means that if they have similar parts across these sequences, those parts are more important than the other parts, the parts which are conserved. So, so they do the sequence search, and then they run a multiple sequence alignment to make sure that the parts which are similar, they are aligned with each other in the columns, right? So this is what MSA is, and you can see that this was maybe the input sequence was from human, but you got sequences from different animals as well. For example, hemoglobin will show up in all the animals. It may look a little different in sequence, but it will have a similar structure eventually because it is performing the same function. So this is one part. Second part they do is they create something called a pair representation. What that means is that for, so the protein is a sequence of amino acids, right? It takes, it creates a pairwise matrix between all the amino acids. So the x-axis is also amino acids, the y-axis is also the same amino acids. It creates a matrix which says what is the interaction between this amino acid and this other amino acid. So that is that representation, and you can imagine that this would be useful when you are predicting a 3D structure, because what amino acids come close to each other and what are far from each other, that is important for the 3D structure. So this is what they're making, and they also do a structure database search. So the sequences which are similar to the input sequence, for those sequences they search in the structure database, get the structures, and those structures they use them as a template. So it's a template of the 3D structure. So you already have some prior information that the 3D structure is going to look something like this. And from this, they create that contact map or pairwise interaction, and these two things they use to create the final pair representation. So this is pre-processing. After the pre-processing, they use this module, evoformer, to refine the MSA representation and the pair representation, and refine it to a point where in the end the hope is that it is encoding the 3D structure, the information for the 3D structure. And the idea is that they do these three things. First is a row-wise attention, which is mentioned here. A row-wise attention will, so you have a 2D thing here, right? So far, whatever we have learned in LLMs today, we just had a one-dimensional thing, and we were attending to different parts of that. Now we have a 2D thing. What do we do here? So we attend in the row level, so we make it one-dimensional and attend in each row separately, and then we attend in each column separately. And the idea is that after you do these two steps and go through multiple rounds of these, you would have accumulated information from different species and different positions in my protein sequence, right? And then the third thing they do is they're trying to make sure that the structure information or the pair former information, pair representation here, which is sort of encoding the structure information, that is a feasible structure. So for example, they want to make sure that the triangle inequality is being followed. So what does the triangle inequality says? That if there is this edge or this bond, let's say in the protein sequence, and this other bond, then this bond cannot be bigger than the length of these two, right? It's not possible. But how do you encode this information in a deep learning module or in a transformer module? So it's sort of doing same thing, which same kind of idea what we did for transformers otherwise. So what are we doing in transformer? We are making sure that we have a mechanism to have the information flow from different tokens to a particular token, right? That mechanism was that query and key and value, that kind of mechanism, but it's just a mechanism. It's not that a token is actually asking a question, do you have relevant information for me? And then this guy is responding that yes, I have relevant information, come talk to me. It's not really happening, right? We're just saying I generate this matrix, and you generate this matrix. If they are aligned with each other, most likely you have some information for me. And that you're providing a mechanism for information to flow from there to here. And when you train this on data sets, that information actually takes the form of, yes, I'm asking for the relevant information and you have that relevant information. So similarly, what they did here was, they developed a sort of triangle attention module where they made sure that the information can flow between edges. So if there are 3 edges which are in a triangle, they made sure that this edge, information of this edge, or the embedding of this edge depends on the embedding of these 2 edges. So there is a way for information to flow from these 2 edges to this edge. And the hope is that as you train this on more data, and you train this making sure that the final outcome, you're testing it for real structures, it will automatically encode the triangle inequality or other such physical rules into the structure. So these 3 things they do, and they go through multiple rounds of that. So you can see that there are 48 blocks like these. In the end, you have a refined MSA and refined pair representation. And then you have a structure module. So you had this MSA representation. Now you just take the first row, which was the actual sequence you had, corresponding to that, whatever embedding you have now. You take that, and that is here, and you have the pair representation, and then you pass it through the structure module, where the key thing is that this IPA module. And IPA stands for invariant point attention. The idea is that this module is invariant to rotations or translations of the structure. So if you have a structure, if it rotates this way, the structure hasn't really changed, right? You just see different coordinates, but the structure is still exactly the same. So you need a module which does not change when you change the orientation, or if you move things like this. So that's what this module does. It does it by doing attention in using relative positions instead of absolute coordinate positions. And then it also predicts the rotations and translations. It also predicts the torsion angles. So once you have the backbone, apart from the backbone in the structure, there are other additional things which are there, and those are called side chains, and there are so those side chains are connected to this main backbone. So if this is my side chain, this is connected to the main backbone. It can be in any orientation, right? So I have to predict the angle from this where it is. So that is a torsion angle. So it predicts that in this final module, and then it outputs the structure. So apart from AlphaFold, there was another model which came out roughly at the same time. So AlphaFold was published in July 2021. This one was published in August 2021. This is from David Baker, who shared the Nobel Prize with the AlphaFold authors. And many ideas are similar to AlphaFold. And in fact, AlphaFold paper got published in July 2021, but the paper was out in bioRxiv much earlier. So they took many ideas from AlphaFold. They wrote that in the paper, that they took many of the ideas and tried that on their technique. And they did one key thing which was different. So in the previous one, if you see, let me go here. In the previous one, if you see, here the first phase is pre-processing. Then you are working with single dimension relationships. And here you are working with 2-dimensional relationships in the form of pair representation. And then in the structure module, you are working with 3-dimensional relationships. So there are separate phases for these 3. But in Rosetta Fold, what they did was, they have this phase where they are doing all 3 simultaneously. So this level is sequence to sequence, so it's 1D. This is contact information, pair former information, so this is 2D, and this is a 3D thing where we had that IPA module in AlphaFold. So they do all 3 simultaneously, and there's an information exchange between all of these. So their hope was that this will lead to a more accurate algorithm. The jury is still out. Here is the comparison. But of course, in deep learning, because data can be different, how data is cleaned can be different, how long you train for is different, your architecture size could be different. It's very difficult to know whether an approach is better or worse. So AlphaFold actually beats Rosetta Fold, but Rosetta Fold is a close second. But you don't know whether that 3-track thing actually helps or not. So we covered 2 protein folding methods, and you can see that there were elements of LLMs for bio in them. We were using transformers also. but they were not what you call a foundational protein language model, because you cannot take that and then say that, okay, now fine tune it for some other task, right? That is, it is trained for a specific task. So for that, there is this model called ESM2. It stands for Evolutionary Scale Modeling. It's a mass language model for proteins. It uses BERT-like architecture, so like we had for DNA BERT and DNA BERT2 and all of those. Its tokenization is amino acid level, and number of parameters. It has models with number of parameters all the way from 8 million to 15 billion. It's trained on 65 million unique sequences from these data sets, and it's trained for following downstream tasks. So it also has a folding solution, which I'll talk about in the next slide, and it covers various other tasks like inverse folding or sequence generation or sequence scoring. So here is the folding method based on ESM2. So the key thing that is different is in AlphaFold or even Rosetta Fold, you have to do that pre-processing part, right? That pre-processing is actually pretty time consuming. It takes 30 minutes to do pre-processing for one sequence, and many times the rest of the part takes maybe a couple of minutes, sometimes 10 minutes depending on the length. Sometimes it may take hours also, but in many cases it takes less than the pre-processing. The pre-processing has been turning out to be a bottleneck. So to address that, what ESM2 did was they learned a foundation model, and then using that foundation model, they can now generate the MSA structure and the pair former structure from the foundation model itself. They don't need to compare with other sequences because the information of other sequences or evolutionary information is already embedded in the foundation model. So yeah, so basically all the steps are the same except the first part, pre-processing, gets replaced with an ESM2 based method. And then in the folding trunk, instead of a full MSA, which had multiple organisms or sequences from multiple organisms, here you will have a single sequence there in MSA, and similarly, and hence a single embedding, 1D embedding thing here. But rest of the stuff is the same. And in fact, the structure module is quite similar to AlphaFold 2's structure module. Then we also have AlphaFold 3. This came out last year, and it is by far the most impressive model. It not only does protein folding, it now can do a lot of other different kinds of tasks. So imagine a biomolecule. A biomolecule can be DNA, RNA, proteins, ligands, which are drug molecules binding to proteins. All of those could be there. And basically, what is a protein folding algorithm? A protein folding algorithm is modeling the interaction between various amino acids in the protein, right? What are the bonds, or what are the affinity between them, or what are the repulsive forces or attractive forces between them, and through that it creates the structure, right? So if you are able to model that for all kinds of biomolecules, then you can do it for anything, right? So you can do RNA-RNA interaction, you can do DNA-DNA interaction, you can do DNA-protein interaction, protein folding, multiple proteins in a complex, you can do protein versus ligand. All of that AlphaFold 3 can model now, and it provides SOTA solutions for all of these categories. The key thing that has changed from AlphaFold 2 is this diffusion module. So instead of the structure module being transformer based, it is diffusion based. It's beyond the scope of this lecture, so I will not go into details of that, but you can read it up in the paper. But it's a fairly different kind of an approach to solve this problem, and it is expected to be more effective in a 3D space. And a few honorable mentions. So there is also ESM3. I talked about ESM2 earlier, and this is by far the biggest model ever trained, protein language model, foundation protein language model ever trained. It is claiming to simulate 500 million years of evolution with a language model. It is trained on 2.78 billion proteins, 777 billion unique tokens. It has 98 billion parameters. It reasons over sequence, structure, and function of proteins. It can do all of these downstream tasks. So it's again one of the most prominent, or the most prominent protein language model right now. Then we also have bolts. So the interesting thing about AlphaFold 3 is the model weights are not public. So sort of AI for bio is following the same thing which AI is following. Earlier, when the models were not as good, people wanted to collaborate with each other. So models were public, people were working with each other, they were building on top of each other's models, and gradually things were improving. Once models got to a point that they were of some value, monetary value, they all became closed source. Same thing happened with AlphaFold. So AlphaFold 2 is public, but AlphaFold 3 is completely closed source. Model weights are not available. Model inference code is available, but without weights you can't do anything. It is available for academic use, so you guys can use it. I can't. DeepMind is directly giving it. So DeepMind is the company which has developed these models. They are directly giving it to another subsidiary of Alphabet. So Alphabet is the umbrella company, right? So it's called Isomorphic Labs, which is a AI for bio startup, right? So they are going to be using that model, and they are going to provide service to everybody else using that model. But it's not available for commercial purpose otherwise. So now, what do people do? So just like there is a ChatGPT, there is also a Llama, which is open source, where there is another entity which is trying to make its name in the world, and because they are not the best, the only way they can make their name is by open sourcing their model and get the community to work on it. Similarly, there are academic institutes which want to continue working in this field, and they cannot do it without collaborations and without community. So there is this group at MIT which has built this thing called Boltz-1, and their goal is to democratize biomolecular interaction modeling. So their algorithm, weights, code, training, inference, everything is publicly available. You can use it, and they achieve accuracy fairly close to AlphaFold 3. Here is just a glimpse of what kind of applications this enables. So you can discover new drugs. I already mentioned AlphaFold 3 can do molecular docking. So if you have a protein here and you have a drug molecule, you can figure out how they dock with each other. It can find out effect of genetic variants. So if I suppose change this letter, I can get the corresponding structure and see if the structure is different and whether it enables different kinds of functionality. It can model protein-protein interactions. It can also engineer artificial proteins. And there's a huge research going on in the artificial protein space. I'll talk about that in a later slide. So challenges and future directions. And I won't spend much time here because they are sort of similar to what we did for LLMs for DNA. Data bias, interpretability, computational cost, these are the challenges. Future directions is, again, multi-omics integration. An interesting area is de novo protein design and understanding of complex biological systems. So before I end, I just want to leave you guys with this very, very interesting slide. So I just took a screenshot from a YouTube lecture by David Baker, who's one of the Nobel laureates along with AlphaFold 2. And he is building a whole suite of tools to design proteins, to design new proteins. And you can see he can design proteins which can create all of these different kind of structures. This was unfathomable for me just like a couple of years back before I saw this, that you can have proteins of these very nice geometric structures also. And what he's saying is that he wants to create this thing like you are playing with Lego blocks. You have various blocks and you can say, how do I combine these together to make this? And now can you give me a sequence which will generate this protein structure? So this is very, very interesting, and this has immense number of applications. It can be used in protein therapeutics, obviously, but even for environmental remediation. So one thing they have been saying is that they want to create enzymes which will just eat CO2 from the atmosphere and output oxygen. So I don't know if that is going to happen or not, but these are lofty goals which they have set for themselves, and it's fairly interesting to me. Here are some resources for further learning. It's also sort of an acknowledgement. I actually went through all of these lectures and few other and all the papers which I mentioned across the slides to prepare for this. So these are interesting talks for further learning. Also acknowledgements to my colleagues, collaborators, and BDBIO organizers. And let me end here. If you have any further questions, I can take them. I know we covered most of the questions in the previous break also. Okay, great. Thank you so much, everyone. Thank you for your time. Oh, yes.
Yeah. So you mentioned that ESM3 is trained on millions of history of evolutionary history. So my question is that can we use such models to not just not just understand the past, but also to look into future, how the human
Evolution will
yeah,
go.
for let's say after 200 or 300 years.
So, firstly, let me clarify what that comment means. It's not that they have taken 500 million year old protein sequences and trained on that, right? What they are claiming is that they have covered so many different kinds of proteins, and in the proteins, it's embedded, the information of last 500 million years of evolution is embedded in the proteins of today, right? So when you learn across all the proteins, you are sort of learning evolution for the 500 million years. But I don't think it can directly be used to say what will happen in the future. But I think a more interesting thing in our lifetime would be where this artificial evolution which is going to happen, where you are going to design new proteins.
Okay.
I don't know if that answers your question, but that's what I know.
Okay. Thank you.
Any other questions?
So post-translational modifications are one big component of protein structure, right? Do these models incorporate that? I don't actually know. Like, is it only amino acid sequences, or do they also have those the epigenetic, or well, not the amino acid modifications that are those included in these models?
So I don't know for sure, but
Okay.
I would think AlphaFold 3 would have it because they cover all kinds of biomolecules.
Okay.
I don't think AlphaFold 2 had that,
I see.
but I could be wrong.
Sir, before we start with any problem, even if we go for basic machine learning, we try to see is it a prediction problem or is it a problem that can be solved by a machine learning. So are there certain parameters that tell us, okay, this is an LLM problem, we can use LLM for that. And what sense does it make for us now to build foundation models in biology? Because certain models, if we fine tune them, like let's say Llama or other open source model, they outperform models that are specifically trained for that particular purpose. So what sense does it make?
Okay, so your first question was how do we know if
It's
this
an
is a
LLM
problem for
problem.
LLMs?
Yeah.
I think a simple solution to simple answer to that question is that if there is a language, inherent language to that problem, and a particular word in that language, and I'm saying word, but you can understand any token or any letter or whatever it is, depends, interacts with words or letters which came before it. So there is some kind of a interaction. Then I think transformers or LLMs can actually model that problem very well. Then we have to see whether it will solve it or not, but I think it will be an appropriate thing to try. Regarding your second question, so this is my belief, of course nothing is proven yet and we have to see, but this is my belief that if models trained on human languages, which are very, very different from DNA or protein languages, if those models are doing as well or better than models trained specifically on biological, that means we have to train on much larger data sets. We don't have we haven't really realized the full potential of those models yet. That's why it's like this. Once we train it on very diverse data sets, large scale models and all that, we should see better performance on this. Because logically that's what seems to be the case, right? If you have something which is trained for some other task and you're being able to use it for this task, clearly the model which you're training for this particular thing or this particular language domain, that is not really done very well. Okay, anything else? Yeah.
The major challenges to develop these models are in plant genomes, because plant genomes are very diverse. Some are diploids, some are tetraploids, and some are polyploids. So this, so while developing these models, so can we separate it through the groups, like separate for tetraploids, separate for diploids, and separate for other polyploids as well?
You can, but
So which will be like it will be like discreting each models.
yeah, you were basically
for-
saying that you will have a plant type based
Yeah.
kind of model, right?
Exactly,
You can
exactly.
do that also, or the other way is that you can develop a foundation model which covers, like you provide data for all different kinds of plants, and you have the model which is big enough that it can encode that diversity,
Okay.
and then you'll have a foundational model which can reason about all of those things.
Okay.
But in the absence of that, if we suppose we don't have the enough data, or if we don't have enough compute to be able to do that, then that's a valid way that you just pick a narrower problem and train for that.
Yeah, because it's very vast, and even
Right.
the genome sizes are also huge for
Also,
some.
we don't have too many plant genomes
Yeah,
sequenced, right?
exactly.
Unlike humans.
Yeah.
Yeah.
Okay. So that was my question. How to?
Okay.
Okay, thank you.
Any other questions? Okay, great. Thank you so much. Thank you for your time.